In this lecture, we will look at what it really means for a drug to be successful. Let's start. What we need to understand is that a drug design is a balancing problem. You will learn the key functions a successful drug must satisfy and why these functions act as interacting constraints rather than independent goals. We will focus on five major constraints. So those are binding, stability, delivery, safety, and manufacturability. So, what makes a successful drug? At a high level, a therapeutic has to satisfy five core functions. First, it has to bind the right target with enough strength to actually have an effect. And it has to remain stable long enough to reach the target. And then it needs to cross the biological barriers between where it is given and where it needs to act. And of course, it has to avoid causing harm. I mean, that is the foundation of safety, right? Finally, it has to be manufacturable. I mean, if you cannot reliably make same molecule, you don't really have a drug. So these five needs that translate into five design constraints we will use throughout this lecture. Binding, stability, safety, and manufacturability. So, and another thing, this is important. So these are not independent checkboxes. So we need to understand that they are all interacting constraints. So let's start with binding. When we talk about binding, there are really three layers to think about. Affinity is about how tightly a molecule binds. Selectivity is about whether it is binding the right thing. And potency is about whether that binding actually matters, biologically. So potency is downstream, right? It is the consequence of binding, not just binding itself. And this is where modeling gets tricky. Improving affinity can hurt selectivity, and potency depends on both. So the goal is not to optimize one metric, it is to find the right balance. Next, let's talk about the forces that drive binding. So when we predict binding, we are really modeling three major type of interactions. Hydrophobic interactions where non-polar regions cluster to avoid water, which we usually capture with log P or log D. And then electrostatic interactions where opposite charges attract and like charges repel, which we estimate using pKa and formal charge. And hydrogen bonding, partial charge sharing between donors and acceptors, which we approximate with the counts of hydrogen bond donors and acceptors and polar surface area. Almost every binding model you build is, some combination of these forces. So for second objective, the key question is simple. Will the molecule survive long enough to reach its target? When drugs enter the body, they are exposed to enzymes in the blood and liver that break them down. So, for any candidate, we look at metabolic stability, usually reported as half life or clearance. Short half life means the drug disappears before it can even work. Too long a half life can cause safety problems. Models predict stability by flagging reactive groups and labile bonds, and by accounting for molecular properties like polarity that influence metabolic exposure. So you will notice some of these same concepts come up again in ADME. Here we are using them to ask a different question: how intrinsically stable the molecule is before worrying about where it goes in the body. So the goal here is not just to make a molecule that binds well, but one that persists long enough to matter. Now we will see objective 3, delivery. So delivery is about whether the drug can physically reach its target at meaningful exposure. So absorption asks whether the drug can enter systemic circulation, which we typically model using solubility and permeability. And distribution describes where the drug goes once it is in the bloodstream, often summarized by a volume of distribution and plasma protein binding. And then the metabolism captures how quickly the body chemically or enzymatically breaks the drug down, commonly measured as intrinsic clearance. And excretion reflects how efficiently um, the drug is ultimately removed from the body, which we usually summarize as overall clearance. So together, these ADME processes determine whether a molecule that looks promising on paper can actually reach its target in a real biological system. And then now we can see objective 4, that is safety. So safety starts with a simple question: does the molecule harm healthy tissues such as heart, liver, or some other critical organs? From a modeling perspective, safety is not about making a perfect yes or no prediction. It is about estimating risk and removing liabilities as early as possible. So specific toxicity focuses on known organ level liabilities. A common example is human ERG inhibition, which signals potential cardiac risk. Liver toxicity markers are another frequent focus. So, whereas general toxicity looks at non-specific cytotoxicity across cell systems, often using measurements like IC50 or the CC50. from cell viability and cytotoxicity assays. So these help identify compounds that are broadly harmful to cells. Structural alert models take a different approach. Instead of relying on biological assays, they flag chemical motifs that are historically associated with risk, such as reactive functional groups. So one important thing here is, paints pan assay interference compounds. So these are often discussed under safety, but um, their primary issue is assay interference rather than intrinsic toxicity. So they generate false positives by interacting with the assay itself, not the biological target. From a data perspective, this introduces misleading labels into screening datasets. Ultimately, the goal of safety assessment is risk reduction, identifying and removing liabilities early, long before a compound ever reaches clinical testing. Now, let's discuss next objective, that is manufacturability. So a molecular design only matters if a chemist can actually make the compound. So the first question is synthetic feasibility. Can we identify a viable route from available starting materials? So modern retrosynthesis tools attempt to explicitly construct plausible synthetic pathways, which is a step beyond older metrics like SA score that only estimate difficulty. So we may also consider, predicted reaction yields when prioritizing candidates. The next question is formulation. Even a synthetically accessible molecule must be soluble and stable enough to be formulated at useful concentrations. So that is where predicted aqueous solubility and formulation stability become important. And finally, manufacturability is about consistency at scale. Can we reliably produce the same compound with the same quality, batch after batch? Models help here by flagging impurity risks and likely side products early. Ultimately, a drug is only successful if it can be produced reproducibly at scale. Now, let's see the summary for this lecture. A successful therapeutic has to balance five interacting constraints: binding, stability, delivery, safety, and manufacturability. Um, no single property can be optimized in isolation. Improving one, often makes another worse. Successful drugs live at the optimal compromise, that's the sweet spot. Thank you.